Yes, we are live.
Not as yet.
It said live button is on.
Now it's there.
Ready to
Yes.
go. Momitash, Professor. Professor Dash, Professor Momita Dash, are you there? Professor Dash? Shomshpro, we are ready to go. It's live, we can go. So, I can't find Momita Dash. Professor Dash, I can see us. Yes, she's there. Yes. Professor Dash, over to you. Okay? You have to unmute yourself.
Okay.
Hello. Professor Momita Dash. Hello, Professor Momita Dash.
I think she was squeezed out. I can see her screen, but I can't see. I think she's back. Professor Dash, you have to just unmute yourself. Just unmute yourself. Professor Dash, you have to unmute yourself again. There's some sort of a technical glitch that we're facing. Kindly unmute yourself. We'll be able to hear you. Yes. Now your microphone should be in a good place to start.
Yes.
Yes. Sorry for that.
Audible?
Yes.
Yes. Okay. So, good evening, everyone. A very warm welcome to all of you to this webinar organized by the Department of
Sir, Momita is facing some network problem, and Fatima will take over from here as Momita's network is not working properly. So I am asking Fatima ma'am to take over the meeting from here.
Yeah, please go ahead. It's already delayed. Yeah, go ahead.
Yes, hello and good evening, everyone.
Good evening.
Without much ado, I would like to welcome all the participants and the audience to this webinar. And I'm sure you all are aware of it, that please keep your audios and videos off throughout the webinar. I would request all the participants. And without much delay, I would request the speaker and first of all, Honorable Vice Chancellor, sir, to welcome everyone, to welcome the speaker. Sir, please welcome the speaker, sir.
Good evening, everybody. Today, it is very important that the Department of Nutrition and Dietetics is organizing this webinar with Professor Dr. Anandita Ray Chakraborty, whom I know for a very long time. And I'm very happy to see her because she almost single-handedly built up this department in Kashishwari College. And Anandita, I find that the topic you have identified is really very, very important. And thank you for joining us. Now, just to introduce the subject, I just want to mention that to everybody who are present over here, that this nutrigenomics is a very important area. And so today we are going to hear from Anandita about this. Next slide. And in case of this nutrigenomics, you know, that the whole concept actually developed after this completion of the Human Genome Project and subsequent identification of the single nucleotide polymorphism. You know that the first thing what is developed is a prediction of individual response to drugs, the pharmacogenetics, which leads to the concept of personalized medicine. You can see the DNA is there. The same drug for a couple of population is safe, and another population it's not safe. Similarly, in certain cases, even it can be unsafe, not effective. And in certain cases, it is unsafe but effective. So all different types of things are possible. Next slide. And there actually started, next slide. So there actually started what we can see, the scope of nutrigenetics. Now, nutritional genetics is a recent offshoot of this genetic revolution that includes nutrigenomics and nutrigenetics. Today, we are going to hear nutrigenomics, but we should also know about its difference from nutrigenetics. Now, understanding this nutrigenetics application is based on the development of some very important cell culture or from transgenic mice or from human intervention, whereby it is possible to do the different types of analysis, which we call as the transcriptome, proteome, metabolism. And the study of the differential transcription leads to the signaling pathways. You know, recently we heard about this COVID pandemic time. We have a huge amount of signaling, which ends up with cytokine storm that led to death of the patient. And all these things are definitely have got some sort of effect on the nuclear transcription factors or things like that. And there are mechanisms of nutrient regulation of metabolism. This nutrient regulation of metabolism is very, very important. And the student of nutrition dietetics, definitely along with biotechnology, microbiology, or in health sciences, they should know about it. Next. So when we find that this popularity of nutrigenetics, genomics, we could see its application in anti-aging, in obesity, as well as in case of the biomix technologies. Next. And when we talk about this anti-aging thing, we know that aging of cells that occurs due to the accumulation of the excess free radicals and formed due to the lack of proper nutrition to the cells. And it is possible to find out how to eliminate the excess free radicals present in the cells by choosing the right composition of nutrients. Next slide. Similarly, if we talk about obesity, which is also the most widely studied topics in nutrigenomics, it is related with hypertension, type 2 diabetes, dyslipidemia, the mood disorders, reproductive disorder, liver diseases, heart diseases, all the different types of things. And they're one of the very important factor, what we call as a body mass index, is very important to do. So it is very important to find out the relationship between the dietary pattern and the genetic factors. Next. And thereby actually identifying the personalized nutrition also. The recent advances in nutrigenomics owed to the completion of Human Genome Project, and it is possible to do it because of the recent advances in our understanding of the omics biology. Next. Next. And thereby, this omics actually what we call as a biomix technology, which helping us to understand this nutrigenomics in a very big way. Next slide. But definitely there are ethics in nutrigenomics, like the privacy, the medical claims, and also the validity of the nutrigenomic tests. And as nutrigenomics is a study of interaction between one's genetics and nutrition, it is very important to do these nutrigenomic tests. Next. So there are a number of rationales in nutrigenomics, nutrients as sins as signal. It is hoped that by building up knowledge in this area, it is possible to promote how nutrition influences the different metabolic pathways, as well as the aims of nutrigenomics also includes being able to demonstrate the effect of bioactive food compounds on health. And I'm sure Professor Anandita is going to cover all these things, because the topic is the story of the food-gene interaction, which is very important right now to understand. Next slide. So I shall end with my lecture, because as I started with, we have to understand the difference between nutrigenomics and nutrigenetics. The nutrigenomics attempts to study the genome-wide influences of nutrition, which aims to identify the genes that influence the risk of diet-related diseases, whereas nutrigenetics examines the effect of genetic variation on the interaction between diet and disease, or on nutrient requirements. So that is the basis of the difference between the nutrigenomics and nutrigenetics. With this, I shall go to the next slide, where I just wanted to thank everybody that you are participating in the seminar, and you will learn quite a bit, but try to get those things, get your questions ready also, and try to find it out. What is actually important lessons, you will get it from Professor Dr. Ray Chocolate. Thank you.
Thank you, sir. Thank you very much for giving the audience a brief overview, a bird's eye view of nutrigenomics. I would like to take a few minutes, dear participants, to just introduce Dr. Anandita to you all, just a couple of seconds. Dr. Anandita is an assistant professor and the head of the Department of Food and Nutrition of Maharani Keshwari College, which is affiliated to the University of Calcutta. She has completed both her graduation and post-graduation from Calcutta University, and she's a gold medalist at both the undergraduate and postgraduate level. She has completed her PhD degree also from the University of Calcutta and works extensively on diabetes and medicinal plants. She has authored 4 books on food and nutrition and has several nutritional and international publications in several international journals. Besides being a prolific speaker at several radio and television talk shows, Dr. Ray is also involved in community-based projects and is an invited expert in several government and non-government organizations. She is an advisor and consultant nutritionist at many food industries and institutes. So, ma'am, over to you now. We are really looking forward to hearing from you on nutrigenomics, the story of food-gene interaction. We are really excited to hear your talk. Thank you, ma'am.
Well, thank you so much for this wonderful introduction. I hope I'm audible. Ms. Kalkatawala, am I audible?
Yes.
Yes. First of all, namaskar and a very good evening to everyone. Speaking after Professor Chattopadhyay sir is a really difficult task, especially someone who has been an inspiration for me all through in my student's life. As he said, I know him from a very long time, and he has truly been an inspiration, sir. And I'm sure, like a true teacher always does, he has done this for me today as well. He has paved the way already for my lecture. Thank you so much, sir. You've already created the ground for my talk. I would not say a talk, maybe a discussion on nutrigenomics. And so with your blessings, I'm sure you will guide me if I slip at any point of time, as you always have. So I'll just take a moment to share my screen. I think, yes. Is it visible?
Not yet.
Is my screen visible?
Not yet.
Yeah. Oh.
Yes.
Yes. Okay. So yeah, I can start. So with due permission from sir and from all the faculty members, I would like to begin by telling you a story. As I said, it's a story, more an epic actually, like the Ramayana and Mahabharata has a lot of characters, different characters. So my story will also have a lot of different characters, where all the nutrients are the characters, because the food that you consume every day, think about the lunch that you had, or maybe the dinner that you are going to have, all of them, just take a moment and think of food as a combination of a lot of different chemicals. Yes, we eat food because of its taste, undoubtedly. We eat food because we like to eat food. Yes, but food is much, much more than that, as sir was saying. And remember the epics that I was talking about, all the different characters play different roles, all separate roles. The genes in our body, the DNA, the RNA, and so many more things that we will come up with. I'm sure there are a lot of young students who probably are new to genetics. I will slightly discuss on that as well. So when they all play separate roles, but when they come together, a new epic, a new story is formed. With this, I would go on to my next slide. Now, what is common to all these people? I'm sure most of you know these figures, I'm sure. Vishwamitra, Parashuram from the Indian mythology, and Hercules from the Greek mythology, of course. So what is common to them? If I might ask a question, I wish I could get an answer, but from a virtual audience, it is difficult. Let's say the word that connects them all is nutrigenomics. Yes. Nutrigenomics is ancient. Now, how are they connected? Shotaboti and her mother, those who are interested in mythology might be knowing the story, were given 2 bowls of porridge with different combinations of nutrients. Obviously, the stories vary, but the most common one is like that. But what they did was they exchanged the bowls of the porridge. So Shotaboti actually gave birth to Jamdagni, who gave birth to Parashuram, who became a warrior from a Brahmin family. And Shotaboti's mother actually gave birth to Vishwamitra, who became a sage, a hermit from a Kshatriya family. Nutrigenomics, maybe because that bowl of porridge that was exchanged contained certain nutrients which might have affected their genetic expressions? A question for all of you. Hercules was given 12 tasks, and one of them was to collect the golden apple that was guarded by the Hesperides. Now, the Hesperides, the word comes from a flavonoid, which is hesperidin, which is present in golden apples, but we do not call them golden apples anymore. We call them oranges. Now, he was asked to collect that, one of the 12 tasks. Why? Because that held the key to good health. So without any further going into the background or the stories, the interesting stories, which might have been examples of nutrigenomics, I'm not sure, just sharing a few examples from mythology where nutrigenomics might have been present. So going into the actual story, as I said, for those who are a little new to DNA and RNA, just to show you how things work to the very basics. Now, we all know that genes present in the DNA coiled on histone proteins, which are again folded many times, different twists and turns. And remember, each twist and each turn has again a story of its own. And believe me, as Sir said, each twist, turn at every organization level, the nutrients can and are playing different roles. As my talk goes on, I will just cite a few examples. Please remember, it is a very complex interaction, but nevertheless, a very interesting one, where there will be several interactions. I will just highlight a few of them. So what happens is, when DNA holds the secret of our lives, we get it from our parents and previous generations, that information is passed on through transcription, process of transcription, as you can see in the slide, the DNA passing information to mRNA, which again is forming proteins through translation. So transcription, translation, and then the proteins that are formed perform several different functions. They regulate several different functions in our body. So you see, nutrients can actually play, I hope the arrow is showing on the screen, nutrients can actually play very significant roles at all the different levels, as we will see some of them. As Sir has already talked about nutrigenomics and genetics, these two pictures can actually show you how nutrigenetics, the genetic pattern can influence how diet is being absorbed in your body, how diet is being changed, metabolized. That is nutrigenetics part. And nutrigenomics is the interaction, the influence that obviously the diet has on your genome. Obviously, these definitions are very simple. There is much, much more to this, I'm sure. But we will not go into the complexities much, but there is obviously a huge difference between the two. So both of them are the two sides of the same coin. Why? Because when nutrients are influencing the genes, the genomic pattern, the expression of the genes, we can also find that the other way is also true as nutrigenomics and genetics. And there is a very complex relation between all of them. And please remember that these can actually make or break a person. By make or break, I mean create health or disease condition of any individual. There might be different types of interactions. There might be direct interactions. There might be epigenetic interactions. There might be variations. And we will just cite a few examples as we go along building up the story. Now, how to study this? How to study this? There are obviously the omics have come into play. When I said DNA, from DNA to RNA was transcription, so transcriptomics. From mRNA to the protein synthesis part is the proteomics. Proteins regulate various metabolic processes, so the metabolomics. So at each level, every bit of it are nutrients. I come from the background of nutrition, of course, so I would be highlighting on the nutritional points. So at each and every single step, nutrition and nutrients will play a role. Please remember, not only the nutrients in the raw state, but we are cooking them. So the effect of every change, every heat treatment, or any spice or anything that you add on food can actually change and influence the food molecule. So whether you're steaming a food, whether you're frying it, that also is very, very important. Now, as nutrigenomics and nutrigenetics interaction is there, we can actually call nutrigenetics as a retrospective science, because it is already there. It's there in our genes. It's a retrospective study that we can do. And a nutrigenomics is a prospective science. Now, what is prospective science? We are having the food now, and that is actually influencing, that might influence our genetic makeup, the expressions, and all the different things. So the nutrigenomics, a retrospective study, nutrigenetics, a retrospective study, and nutrigenomics, a prospective study. So keeping this in mind, we can obviously understand the principles, basically, of nutrigenomics. Now, what are the principles? We can see it here. So as we already mentioned, that they are dietary chemicals which alter gene expression, might alter, I would say. So improper diets would obviously alter it in a very different way. So that is a risk factor. Then there would be influences on genes. There would be influences on the disease patterns that we will gradually unravel as we go about. As Sir had already mentioned, SNPs, I will not go into the details, but inborn errors of metabolism like PKU, I've just mentioned two of the most common ones, that PKU and lactose intolerance. Now, PKU is phenylketonuria, in which phenylalanine is not properly metabolized. So phenylalanine hydroxylase enzyme, again, a protein, which is regulated by, obviously, translation and transcription through your gene. So that enzyme, if is not properly produced, a person will get PKU. And if it is lactose intolerance, the enzyme lactase, again a protein, so proteomics and metabolomics being influenced, this is actually influencing our dietary pattern. So lactose, a nutrient, phenylalanine, a nutrient, these are not being properly metabolized because of an inborn error where there's a faulty gene that is not letting us synthesize proper enzymes. Now, there is also difference in sex-based differences. There are a lot of journals, and if you just go through them, if you're interested, you can see various sex differences associated with nutrigenomics as well. But I've not gone into that detail particularly. But there are various ways in which males and females have different metabolic patterns. So you will see some kind of diseases very common in females, some kind of diseases very common in males. Obviously, there will be some differences that can be explained on the basis of nutrigenomics. So coming to each example now, one after the other. I will obviously not go into the complex mechanism, because I was not too sure whether students were there who know a lot about genetics. So let's just go from the nutritional point of view, that is nutrigenomics. How? We all know folic acid and vitamin B12. What do? They transfer a single carbon moiety as in a group, methyl group, from one substrate to another. You can see on the slide. And they can, for example, they can interconvert methionine and homocysteine. Now, when this interconversion takes place, this actually helps in DNA methylation and inhibits the gene expression. So you see folic acid and vitamin B12. Those studying nutrition would just remember them as factors, nutrients, regulating anemia, relating to anemia. But please remember that it is extremely important, and how it is important. It helps in DNA methylation. You can also see the HAT as in the histone acyltransferase proteins, which are also there, the enzymes, which can be manipulated, which can be regulated with the help of these nutrients. You can see so many curcumin present in turmeric, holud, as we call it, resveratrols, catechins, epicatechins. These are all extremely important nutrients. I would not say nutrients, phytochemicals, which can help to regulate the expression of genes, which can help to regulate how your DNA functions. Remember, we are born with a certain set of genes, but how we go about it, there can be some changes depending on your diet, and that is what we are discussing. So after folic acid and B12, vitamin B12, how can I not mention breast milk? Because this is the World Breastfeeding Week going on. I'm sure most of you know, from the 1st to the 7th of August. So breast milk from the mother, where it all starts. You see the diet of the mother. You can see here the PUFA, MUFA, the trans fatty acids. They can actually regulate the transcription factors like the PPARs or the peroxisome proliferator activator receptors and the SREBs. So what happens to these? These are the transcription factors which actually regulate the genes. You can see here, FADS1, FADS2, sounds complicated, but they are not. They're actually fatty acid desaturase enzymes, which can, the genes which control these, which can actually carry out fatty acid synthesis by desaturation, and ELOV2, ELOV5, which are regulating the elongase enzyme. Now, these regulations can actually help in the production of the kind of fat present in breast milk. So you see, diet of the mother influencing the transcription factors, and transcription factors regulating the production of enzyme proteins, which can actually change or alter the proteins or the type of fats present in the breast milk. And this breast milk is the food for the next generation. So an infant would be regulated having this particular set of nutrients. So you see, it is miraculous. I have just highlighted one interaction. There are several. In fact, I don't know, it's actually innumerable. I sometimes get so excited speaking on this. I don't know whether I'm being able to convey to you how important this particular factor is. So this is just about fat. Please remember, there are proteins, there are carbohydrates. The amount of carbohydrate that a woman consumes during the lactation period, during her pregnancy period, they are all very important in terms of nutrigenomics. If anyone is interested, I have cited the references also, so you can just go through them if you want. Now, talking about a vitamin which is no longer a vitamin. We call it a hormone, yes. So vitamin D now behaves like a hormone. Not now, it actually behaves like a hormone. We generally call it or refer to it as the sunshine vitamin, though it is basically a hormone-like substance which can regulate a number of functions in our body. And it is known as a hormone because it can actually regulate the functions of DNA like hormones do, fat-soluble hormones do, like the steroid hormones. Now you see here, you can see the pathway that takes place. The cytochrome P450, the family of enzymes, can regulate hydroxylation of vitamin D. In the liver, the vitamin D is hydroxylated at the 25th position. Then it goes to the kidney for 1 position hydroxylation and again a 24 hydroxylation in the liver. This goes on. And due to this hydroxylation reactions in the liver and the kidney, vitamin D gets activated. And this activated vitamin D can bind to the VDRs or the vitamin D receptors, which can, in this slide, you can see, this can regulate the genomic expression, the genomic effect. So vitamin D being regulated by hydroxylases, which again are controlled by the genes because they are proteins. And this vitamin D in turn is again regulating another gene, genetic makeup, where we can change the expression that takes place. So it is unique and a both-way action taking place all through. Now, zinc is again it's a very controversial mineral, a nutrient, which is being studied extensively nowadays. because zinc is found to play very, very important roles, not only in nutrition, but also is an important part of genetics as well. For those of you who know, zinc forms a core element of the hormone insulin as well. So a lot of research is being also done on diabetes, insulin, and zinc, but I will not go into that detail right now. So zinc ions are needed for RNA polymerase activity. So RNA polymerase, we all know what it does, building up the molecules. Actually, polymer, it is a polymerase, so it will polymerize the small particles and make up the whole molecule, RNA molecule. Now, zinc finger proteins, here you can see the zinc finger proteins getting attached to the gene and regulating as transcription factors. They behave like transcription factors and actually carry out protein-protein interaction in kinase binding proteins. Now, what are kinases? Most of us know that kinases are the energy equilibrium maintaining enzymes, ATP, ADP balance and all that. It's a biochemical concept where kinase can convert ATP, ADP. I'll not go into that particular detail, but it is obviously a very important enzyme that helps, that is being regulated by zinc finger proteins. Obviously, zinc is the important nutrient here. Now, zinc can directly regulate transcription rates as well, directly regulate the transcription rates. Just imagine a food that you're eating, for example, maybe a sea fish or something, or a sesame seed containing something, tile naru, very favorite of us Bengalis. So if you have that, has a lot of zinc, that zinc digested, absorbed, and then going into the makeup of the changing of the gene, the genetic pattern, and regulating so many fantastic metabolic pathways in our body. So you can see how directly the regulation takes place and how physiological modifications also take place. Now, after the nutrients, just to give a recap, everything has a recap nowadays. So to give a recap, folic acid, B12, vitamin D, we had talked about, zinc we had talked about. So all these are the nutrients which are important and being studied from the nutrigenomics point of view. Now the diseases, I'll just talk about 3 diseases where these nutrigenomic reactions are taking place. So the first one, as sir already mentioned, obesity. Very complex picture, very, very complex picture. Just one interesting interaction that I will talk about, just one. Please remember, there are probably, sir would actually be able to tell me exactly how many. I'm not too sure as how many interactions are there, but I'll just highlight one. That is, those who have this particular allele, 11391A allele, they have a significantly lower risk of obesity, even upon the consumption of fats in the diet. Wow, they're lucky people, I must say, because if eating fats do not lead to obesity for these people, I think it is quite a blessing. There are also carriers of adiponectin and several other factors which can actually control how fat is metabolized in your body, how fat gets deposited, the adiposity, and how or whether you will become overweight, underweight, or obese. As I mentioned, this is just a glimpse into the complex picture of obesity. You can see at all the different levels, the genetic makeup, the epigenetic patterns, the gene X and the environment interaction. This is something which we nutritionists are extremely interested about, the gene X environment interactions. So this is basically a peek, just a small peek into the myriad world of nutrigenomics and how it relates to disease. As I was saying, complicated. See how complicated it is. Just see the interactions. Just go through these interactions. Nutrients, food groups, the dietary pattern. So it might interest you to know that the time when you are eating food, there are reported values as to how these actually control the metabolism and how these control the gene expression. It is really fantastic. And a 45 minutes lecture obviously is not enough to highlight all the points, just to go through the entire thing to make you realize that food is not what it seems to be. So this complex interaction, this entirely complex interaction, where again, just highlighting one, the E4 allele present in the apolipoprotein E gene shows that a higher LDL level in individuals who are eating the same and equivalent amount of fat, the same composition of fat as compared to those having E1, E2, or E3 allele. So these are the unlucky people who have a higher LDL value as compared to others who are eating the same kind of fats. So these are very unique and specific for each nutrient from the point of nutrigenetics as well as nutrigenomics. Obviously, there is some kind of overlap because one, as we saw that they're the two faces of the same coin. And one is a retrospective study, as I said, and the other is a prospective study. Diabetes. As I talked about Hercules and hesperidin, now this is how hesperidin works. Hesperidin is very important in terms of liver glucokinase, obviously a very, very important enzyme for glucose metabolism. There is olenolic acid, there is apigenin, luteolin, and there is glutamine. Now, glutamine is very, very interesting because this particular nutrient can regulate calcineurin, which is a calcium binding protein. And this can regulate the functions of the pancreatic beta cells. There is a very elaborate mechanism which actually takes place. So see how glucose homeostasis is being regulated by so many different nutrients, where nutrigenomics is playing a significant role in the metabolism of glucose. So as we are talking about this, as we are talking about specific interactions, you must remember that the subject of food, the subject of nutrition is very, very specific. So it cannot be just 65% as the nutritionists calculate the diet chart. 65% of calories should come from carbohydrate. No, it has to be specific. It has to be a specific term, a specific it has to be specific for each individual. So to sum up the entire thing, nutrigenomics, nutrigenetics, coming back to the same thing, you can just follow this diagram, how nutrients influence the DNA, the gene expression here, transcriptomics, as we said, proteomics and metabolomics, how the health status and the disease conditions are maintained, how they are also controlled by the SNPs, the genetic variability affecting the nutrients as well. So these are all the various different types of interactions that we have talked about, the nutrients, the disease part, and then the total interaction. Everything will have some pros and some cons. There would be potential benefits of nutrigenomics. Obviously, there will be ethical issues, as Sir said. There would be disadvantages as well. But if we balance them, I think the concept of nutrigenomics will definitely help us to understand how each and every particle of the food, each and every particle of the nutrient can have a bearing on the disease condition of our body. So genetic profiling is definitely the need of the hour if it is possible. If we can carry out this difficult, I understand it is very, very difficult, but it is the future. We will have to talk about genetically tailored therapy, because genetically tailored therapy is the answer to any kind of, especially the lifestyle diseases, be it obesity, be it diabetes, be it cancer, anything, heart disease, as we saw the complex interactions. So it has to be tailor-made, and that is where the importance of studying nutrigenomics is. You can see from here how nutrients genotype 1, genotype 2, and genotype 3. It can actually be different for different people. That is what we saw all along. I just highlighted each one of several interactions, which I thought could make you understand the importance of how each nutrient and how each part of a complicated DNA or chromosome can interact and carry out the whole story. So this is a complete, not a complete, it's not an exhaustive set, of course. There are so many interactions, just a glimpse for all of you to show you how all these factors, the nutrient factors, can affect so many of the genomic particles in our body. And this actually carry out the science, the mechanism of the process of nutrigenomics. Again, to highlight, every nutrient has a different mechanism to play, which obviously is not possible to highlight in such a short time. But together, when we bring them all together, think about a morsel of food that you have eaten in the afternoon, maybe. It contains carbohydrates, all different types of carbohydrates, all different types of proteins made up of amino acids, different types of fats, each of them put together, going into your microscopic cells and the nucleus and the DNA, can bring about a huge change in your body makeup, a huge change in how we stay, how we live. And this, as I said, everything starts from the mother. The mother is the focal point. So we have to be beware even before we are born, because what the mother eats, the diet that can actually regulate. I'm really sorry about the noise, but I hope I'm audible. But the genetic makeup that is there, the change that can take place, can actually help, can actually regulate the genetic makeup of the child, the fetus. And this is very, very important, because this is the set of genes that the child is going to be born with. And naturally, his entire life would be affected by this. So just before I end, for all the young people out there, genes make you what you are. That is, of course, out of our control. But our lifestyle can change it all. And diet plays I left out the one because I am from nutrition. So diet plays the most important role. So this is the plate that you should keep in mind, the precise nutrition plate. So thank you very much, and over to the organizers.
Thank you very much, ma'am, for the wonderful talk. The way you started, ma'am, it really gripped our attention. It was wonderful. And the whole talk was so gripping. We just couldn't, it was really amazing. Thank you, ma'am, for the wonderful talk.
Thank you so much. It's a pleasure.
Yeah, Anandita, you have done a really great job.
Thank you, sir.
From the first slide to the end, we are stuck with all the different PowerPoints and the way you logically developed, and you know that you have covered very important cardiovascular disease as one of your case study, as well as that our lifestyle, how it's going to help not only the pregnancy, it is actually, we can see it's for the next generation, how we are prepared. So we have to think about, we actually can have a lot of different things, whether that is going to affect our next generation or not. So that is also a very nice way you have presented. So I hope everybody who are with us, you understood that what is the importance of the good lifestyle, good nutrition, and trying to be very, very cautious about what you are doing, just only for the sake of fun or anything. So this is a very important lesson we learned from Anandita's lecture. Thank you, Anandita.
Thank you so much, sir. Thank you so much.
Thank you, ma'am. So can we just take up a couple of questions from the audience? Like we have a few questions. Ma'am, if you allow, can we take up a few questions?
Yes, absolutely, if time is permitting you.
So ma'am, one question is from, just one second. Yes, Shwetanjana Ghosh. She's asking how nutrigenomics and nutrigenetics are directly related to the beneficial role of gut microbial niche and what kind of interaction do they share?
I will request sir to answer this because in front of him, I really don't think I should be answering this. Sir, I request you to please take over this question because he is an
yes,
expert.
I think Professor Ray actually mentioned very correctly that the gut microbiota has got a very important role in a lot of different types of disorders. It's not all disorders, but a lot of different types of disorders. And in that case, we have to actually try to find out that the genetic makeup is important. Say in a genetic makeup, in certain cases, some people, some population, a particular dietary or nutrient may not be so much effective with respect to a particular type of gut microbiota, which again considers all the different types of things. So it is a genetics which actually it's a food gene interaction. What is actually the topic today is very important to find out whether the gut microbiota is going to have important role or not. Thank you.
Sir, in continuation with this only, like there is a second question, like they want to know that if a person's genetic makeup and nutrient gene interaction, like knowing that, is it possible to make a personalized dietary pattern for that person?
Is it a question to sir, I think, or should
actually to both of you?
Okay.
Yeah,
so
definitely. That's what Anandita actually told us so far. We learned from her lecture.
Yes, actually, so if I can, with your permission, if I can answer a little bit of this, because see, the genetic profiling here is a difficult proposition. See the population, it's very difficult, I understand. But you see the concept of a generalized diet, 1 gram per kg ideal body weight protein, as I said, so much of fat, this much of fiber, 18 grams fiber, 20 grams, this cannot go on because see, each individual is an individual. So everyone has a different genetic pattern. Everyone has a different requirement. So I understand that we will have to work on a reference man and a reference woman, as ICMR has given us. ICMR, as an Indian Council of Medical Research, has done the diet charts on the basis of reference man and reference woman. Obviously, they cannot do it otherwise. They cannot take each and every person and plan a diet. Not possible. But you see, if that's a dream, if it is possible, if we can, there's a concept called diet counseling in nutrition and diet therapy. So if we can just talk to the person and get the habits, if we can get the timing of food, if we can get the type of disease patterns, and if we can have a history taken, we can narrow down, I think, the dietary pattern. I'm sure all of you from nutrition, all the faculty members would also agree that this has to be the future. It cannot be a generalized pattern. So I think it is possible, yes.
No, actually, I have one comment to make. Like, we don't have something like one size fits all. It has to be tailored.
Absolutely. I completely agree with you.
So, ma'am, actually, if you can take up two or three more questions, like it's from.
I don't mind. I love answering questions. I'm a teacher.
So Dr. Nilanjan Das has one question. That can the study of nutrigenomics or nutrigenetics be helpful to address the days of COV2 induced infertility?
Days of? Sorry, I didn't get you.
Of COV2 induced, the SARS-CoV-2 induced infertility.
Well, I am not too sure about. Can you just repeat the question because your voice is? Sure. Is it written in the chat? So.
Yes, ma'am. It is there in the chat box from Dr. Nilanjan Das.
Oh, you can just repeat it for me because the voice is breaking up. I can't hear. I'm really sorry. Just repeat it. I'll get it.
Can the study of nutrigenomics or nutrigenetics be helpful to address the days of SARS-CoV-2 induced infertility? Now, I can understand it's a very difficult question to answer.
I will have to really look up journal papers relating to this. I am not too sure, but I'm sure there must be some effect. There will be definitely some effect because, as we said, since I can share a few informations if that helps. Infertility relating to vitamin E deficiency, infertility relating to selenium. Selenium and vitamin E obviously work together, as you know. There's an interrelationship. So these might have some kind of relation to infertility, but I really did not understand the COVID and the infertility relation. That I am not too sure as to what Professor Nilanjan was saying.
I had the same thing in mind actually, ma'am.
Yes, so vitamin E and selenium, yes, related to infertility, but I'm not too sure about the other part.
Okay. And ma'am, last two questions. One is from Banishika Roy. The question is, there are many sports person who are not aware of lactose. Just ma'am, 1 second. Who are not aware of lactose or gluten intolerance, and they never felt any discomfort. But when gene testing was done, it was showing that they are having gluten and lactose intolerance. So she wants if you can elaborate something on that.
This is a very, very uncommon phenomenon, because anyone who has lactose intolerance and gluten intolerance will obviously have these symptoms whenever that person is having gluten or lactose. Even gluten in the disguised form. For example, if you talk about flour, atta, maida, whatever, and if you put it on, say, we Bengalis love making chop, cutlet, using that. If you coat it with breadcrumbs or anything that has flour, that person will have symptoms. So it is a very uncommon phenomenon. I've never come across something like this, but I will definitely have, if you can just give me some more information on this, it would be really nice, because this is a very, very uncommon phenomenon that I am hearing. This first time probably.
Okay. Thank you, ma'am. And I'll just take one last question. That's from Biksha Basu. Ma'am, she wants to know what is the major role of nutrigenomics and nutraceutical selection in the management of obesity?
Yes, I think Diksha Basu.
Yes.
See, we were talking about obesity, and as I said that I just showed you one or two genetic makeup, the alleles that can metabolize fats in a different manner. So you see, we will have to understand the genetic profiling before I can predict that this particular thing should be consumed. But as a whole, according to the ICMR standards, if I go with the reference values, we can obviously stick to the general dietary plan for obesity if that person is normal weight. See, the ICMR guidelines always have a buffer. Please remember that ICMR will always have a buffer. So if you can stick to that particular diet, if that person is not already overweight, if he's overweight, obese, we will have to think otherwise. Otherwise, you can stick to all the nutrients that we generally give in the pattern that a normal reference man or woman can be given.
Thank you.
I think I could answer her question.
Diksha? Yes. Thank you, ma'am. Thank you so much for the wonderful talk.
If I can just take a moment, I just saw one question. I'm really sorry about this, but it's a very interesting question between nutrigenomics and mental health. Yes, I wanted to include slides about nutrigenomics and mental health. See, mental health is dependent on neurotransmitters. Vitamin D deficiency has been proved to lead to depression. There are several factors on nutrigenomics which actually regulate the mental health, depression, and is related to suicide rates. So for all the young people there, please look these things up, have the proper diet, have things like dark chocolate, if you like, that can stimulate your brain health and keep you happy and healthy always.
Ma'am, we look forward to another webinar from you on this topic in that case.
I would love that. I would love that. Absolutely.
Yes. Thank you so much, ma'am. And now, ma'am, I would request the head of the department, Dr. Shomshubra Thakur Chaudhary, to deliver the vote of thanks. Thank you, ma'am.
Fatima, am I audible? Okay.
Yes, yes.
Thank you, ma'am. Thank you for the beautiful lecture. It means what Fatima has correctly said, from the first slide to the last. It was extraordinary, and I am really grateful that you have from your busy schedule, you have taken up time so that you can enlighten us. I hope whenever this COVID situation will be over, we can have an offline session also, when you can interact with our students and I think you would love to interact with our students. As I also want to thank our VC sir, Professor Jogojyoti Chattopadhyay. Sir, without your help, without your support, it was very much impossible for us, for a new department to uh, grow up to do such events regularly. And please support us. Please keep your blessings on us as so that
Thank
we can
Thank
grow
you,
more.
Shomshu bro.
I also want to thank the Wendila ma'am, the Ina ma'am for their support to organize this webinar. I also want to thank the whole Department of Applied Nutrition and Dietetics of Sister Nivedita University. All the faculty members of our department, they was very much enthusiastic about this webinar series, and their support, their help is the thing that helps us to go forward. And with this, I want to end my end this webinars of today's webinar. And I want to mention that throughout today's webinar, different questions has come about the personalized nutrition, and I am happy to mention that tomorrow Dr. Dilip Ghosh sir will give us a talk based on this personalized nutrition. His topic is personalized nutrition, so those who are interested can also can be a part of tomorrow's webinar. So with that, I want to ask Fatima to take up the next part of the session.
Yes, thank you so much, ma'am. And we will look forward to future webinars with you, ma'am. Thank you so much. With that, we would like to conclude today's session with the Honorable Vice Chancellor Sir's permission, sir.
Please go ahead. Thanks to everybody.
Thank you, sir. Thank you.
Thank you, sir.
Thank you.
Thank you, ma'am.